Myself, my name is Mark Kiel. I am the Chief Science Officer and founder of Genomenon. Genomenon is an AI-driven genomics company that is saving and improving lives by making genomic information actionable. And I'll speak more on that later. But my background is as a molecular genetic pathologist with both a clinical and a research degree, an MD and a PhD. And in my role as Chief Science Officer, I supervise the strategic direction of Genomenon.
Hey, my name is Catherine Nester. I am the Vice President of Physician and Patient Strategies at Inozyme Pharma. And at Inozyme Pharma, we are developing treatments for rare mineralization disorders. And one of those treatments is in the clinic now. And I'll talk a little bit more about that as we go. The initial prevalence for the disease was thought to be 1 in 200,000 pregnancies. And as we often see with rare diseases, the more we learn about the disease, the more disease awareness is raised. You know, we start to uncover more patients that are diagnosed, and that's led to a lot of work that the Genomenon team has done. And I'm going to let Mark speak to that.
Yeah, sure. That's a great segue. So when you ask me what prevalence is, I might say, who's asking? What do you mean by prevalence? And so that gives me an opportunity to put a little bit more perspective or context on what a prevalence number is for a rare disease. So one of the ways to think about prevalence is what Catherine described as what the actual number of these individuals with this disease is across all individuals in a population. That's a theoretically knowable number, but knowing that number is really challenging. And it's challenged in large part by the rarity of the disease. So we're left with proxies for what that actual number is. And there's two ways to think about those proxies. One is with your clinical experience. The clinicians, the key opinion leaders, the experts in treating the disease, as well as the primary care physicians, how many of these patients are they seeing when they do recognize that they're seeing one of these patients and get that diagnosis confirmed? So there's that clinical intuition around the prevalence, but then there's the evidence predicated calculation of that prevalence number. And the challenge in putting together that number is compromised by the access to and quantity and quality of that evidence. So very often in rare and ultra rare diseases, where the prevalence number is critically important to get accurate and to have evidence predicated around it, there's reliance on insufficient evidence to the point where sometimes there's even just a guess. And so what we're talking about in the context of the collaboration between Inozyme and Genomenon, and Genomenon's efforts to take the fruits of that collaboration and determine the prevalence of the Gacy disease, is applying this more robust, better annotated, more accurate evidence predicate in the service of getting closer to that true prevalence number based on evidence. And that evidence comes from the primary scientific and medical literature and Genomenon's ability to curate that information and then to apply a statistical mathematical model for this autosomal recessive disease to get that precise evidence predicated number. Sure, let me take a step up. Let me tell you what AI-driven genomics is very briefly, and then I'll go into a little bit more detail about how that number was calculated and how it's defensible with that evidence. So what is AI-driven genomics? It is the marriage between human expertise and computational capability for making both more and more accurate genetic diagnoses. AI is a very powerful capability, and it has broad applications in many fields, one of which is biomedicine. And in particular, within biomedicine, genomics is perfect for this type of application. If you think about the past, we have the Human Genome Project that made this possible to sequence patients and understand the causes for any disease they may be suffering at the genetic level. And then we had the next generation sequencing revolution that made that practical, made it possible to get that information very quickly. But now we're talking about making it applicable to the masses, making it more widely available by solving the interpretation challenge. Production of this genome data on a wide scale produces this bottleneck, or the bioinformatic or interpretation bottleneck. And this is a perfect opportunity for AI to prove its mettle. And that's where Genomenon comes in, is organizing the world's genomic information, particularly valuable for rare diseases and cancer, to help uncover all the patients who have been described and all of the genetic variants that have been determined to cause their disease, and organize and annotate that evidence in the service of finding more of those individuals and making more accurate diagnoses with that data. Genomenon is on a mission to curate the entire human genome in this way, one variant at a time. And in the context of the work that we have done with Inozyme for ENPP1 deficiency, we have curated every one of the ENPP1 disease-causing variants, curated it according to a clinical standard framework, leveraging our computational intelligence, organizing and pre-annotating all of this evidence. And in that context, that data is being put to use in the service of helping identify and more accurately diagnose these patients, but also to use that evidence to better predict a more accurate reflection of the prevalence of this disease. So very specifically, the way that we did that is because this is an autosomal recessive disease. That means to say that 2 copies of the disease gene have to be present in the mother and the father, typically. Those mothers and fathers who have 1 copy that is disease causing of the gene, when they get together and have offspring, there's a chance that those offspring have inherited 2 copies of the disease gene variants. And when that happens, then we're talking about the need to recognize those individuals clinically. So given the context of this inheritance pattern and our ability to have understood all of the disease-causing genetic variants based on our curation of the medical literature, we were able to apply statistical models using population databases of healthy, normal mothers and fathers, some of whom are carriers of disease-causing variants. When we put that data together, it allows us to calculate the prevalence, or the number of individuals who are born with the genetics that's appropriate to causing ENPP1 deficiency. So it's important for me to underscore that we use the same methods as had been previously applied in the service of calculating the ENPP1 deficiency prevalence, but also the same methods that are accepted by epidemiologists across the board for these types of autosomal recessive diseases. The difference is that Genomenon, given its AI computational capability, had access to better and more such data. We were able to find more variants, and for each of those variants, more studies, functional studies determining the role of these variants in causing disease, as well as clinical studies demonstrating that these variants do in fact associate with diseased individuals in the clinic. So this is a great realization of the benefit of the AI plus human expert curation approach in the service of identifying more individuals with rare disease and putting a more accurate number as to what the prevalence is for those individuals.